Thank you for viewing Biologics oncology-focused presentation today. I'm Dr. Hal Gunn, founder and CEO of Biologics. Q has developed an innovative approach to harnessing both innate and adaptive immunity and targeting that response to a specific organ or tissue. It's a first-in-class platform with broad applicability. We have compelling data from 4 phase 2 clinical trials, and we have a 5th phase 2 study underway in colon cancer. We have had a very good safety profile to date in the more than 360 patients we treated. We have a robust patent portfolio and a strong, inspired team ready to deliver on the promise of our very novel approach to cancer therapy. We're seeking VC funding and pharma partnership to accelerate clinical development. Q's platform is uniquely positioned in oncology in 7 important ways. First of all, our platform is organ targeted. We can target an anti-cancer innate immune response to a specific organ or tissue like the lungs or the gastrointestinal tract. Secondly, we activate and recruit multiple important immune cells to that targeted organ, activated natural killer cells, M1 macrophages, and activated adaptive immune cells. Thirdly, we activate and restore multiple important anti-cancer immune pathways, including NKG2D, which makes cancer cells more visible to the immune system, and multiple different checkpoint inhibitors that are downregulated to restore both innate and adaptive immune function. As I mentioned, we have a very good safety profile to date. We anticipate a safety profile similar to vaccination. We've demonstrated synergy with a broad range of cancer treatments, including chemotherapy and various immunotherapies. Because of the safety and because our treatments restore normal immune function, we believe we have application in both prevention and in treatment. And our platform can be broadly applicable beyond cancer. We've demonstrated and published in COPD, asthma, Crohn's disease, ulcerative colitis, and infection. The best way to think about our platform is as innate immune vaccines that restore innate immunity in a targeted organ. The basis for our platform is our discovery that the innate immune response to bacterial infection includes an organ-targeted innate immune activation that restores all of the cells and roles of innate immunity in the targeted organ. Because an important role of innate immunity is to modulate adaptive immunity, when we restore normal function of innate immunity, we help to restore normal function of adaptive immunity. As I mentioned, our platform is like an innate immune vaccine. We call our platform SiteSpecific Immunomodulators, and I'll be referring to that as SSIs. They're derived from inactivated bacteria, similar to a vaccine, and they're designed to safely and effectively target an innate immune response to a specific organ. They act through trained innate immunity and organ-specific chemokine release, and I'll be reviewing the mechanism momentarily. And as I mentioned, they restore multiple important anti-cancer immune pathways and cells. They're self-administered by simple subcutaneous injection. And even though they're delivered by subcutaneous injection, they create a targeted response in a specific organ or tissue. And this slide depicts the organ targeting of the SSIs. We've developed 3 SSIs that we've taken into the clinic today. The first derived from E. coli, which is endogenous in the GI tract, and the SSI derived from E. coli targets that anti-cancer immune response, the restoration of innate and adaptive immunity to the GI tract. The second, made from Klebsiella pneumonia, which is a lung pathogen, targets that response to the lungs. And the third, made from Staph aureus, targets that response to the skin, where Staph aureus is endogenous. As I mentioned, the SSIs are made from inactivated bacteria, so they can't cause infection. And even though they're injected just under the skin, they create this targeted innate immune response to the specific organ or tissue where those bacteria are endogenous. There are 6 steps to the mechanism, and I'll discuss them briefly. The SSI is self-administered by simple subcutaneous injection under the skin. That creates the release of cytokines as a danger signal that then activates hematopoiesis in the bone marrow with release of primed innate immune cells into the bloodstream. The SSI is also carried systemically, and it's only in the targeted organ where trained innate immune cells that have seen that bacteria before, because it's endogenous in that organ, release chemokines, CCL2s, CXCL9, CXCL10, that then draw those released primed innate immune cells into the targeted organ to restore innate immunity and restore all of the important roles of innate immunity, including the activation of adaptive immunity and many of the important roles of the anti-cancer immune response. This slide is showing the broad applicability and targeted nature of the SSIs. We are using 4 treatments in each of these mouse models: placebo, the lung-directed SSI, the GI-directed SSI, and the skin-directed SSI. And you can see lung cancer and lung infection, GI cancer and GI infection, and skin cancer and skin infection. And in each case, it's only the targeted SSI that has efficacy, and it has substantial efficacy in the targeted organ because of the recruitment of the activated innate and adaptive immune cells. SSIs induce organ-specific innate immune cell recruitment, including both myeloid cells and natural killer cells. And these cells are activated. Here we're showing that the cells that are recruited to the targeted organ are activated, for example, M1 polarization to create that anti-cancer immune response in the targeted organ. Here we're demonstrating synergy with a wide range of treatments: chemotherapy, sting agonists, and cancer vaccine. And in each case, it is the combination of those drugs that are most effective, even though SSIs have an effect as a single agent therapy. We've demonstrated efficacy in solid tumor models in combination with adoptive cell therapy, including CAR T cell therapy. You can see a substantial synergistic effect. This was in studies in collaboration at Dartmouth College. The important principle here is that SSIs create an organ-specific chemokine release that then draw adoptive cell therapies like CAR T cells into solid tumors. We think it has a really important application in the CAR T cell space. We did a small study in lung cancer looking at patients with recurrent lung cancer who weren't on any other treatments, and you can see a substantial downregulation in PD-L1 expression, a substantial downregulation in PD-1 expression, and we've demonstrated a downregulation in other checkpoint inhibitors. In a compassionate use program, where we treated more than 250 patients with advanced cancer. We demonstrated substantial survival advantage compared to other patients with the same stage and type of cancer treated at the same clinic. These are not randomized controlled trials, but they're suggestive of a strong benefit. This is an example of a patient treated in that program with advanced inoperable pancreatic cancer. After 2.5 years of treatment with SSI therapy, you can see her cancer is cleared. And she is now cancer-free more than 8 years later, which is a remarkable event in advanced pancreatic cancer. You can see in stage 4 breast cancer, we found a 20-month median survival advantage for patients treated at that clinic with SSI therapy. And in a case-matched study across a range of advanced cancers, we found a 12-month median survival advantage. Again, these aren't randomized controlled trials, but are suggestive of significant promise. We have a phase 2 study underway in colon cancer where we're looking at the impact of our treatment on the tumor microenvironment. We've demonstrated in a number of cancers preclinically that our treatment activates multiple mechanisms and shifts the tumor microenvironment from immunosuppressed to immunoactivated. We have a very strong safety profile to date. As I mentioned, we anticipate a safety profile similar to vaccination. We have strong patent protection with 61 granted patents across 5 important patent families. That includes cancer and enhanced adoptive T-cell therapies. We're seeking VC funding and pharma partnerships to accelerate our clinical development. We have a strong team and a strong board of directors who are excited about the potential of our technology in oncology and across a range of chronic inflammatory diseases. We have a strong team of advisors, collaborators, and academic partners. So in summary, we have a broadly applicable platform technology that's very unique and innovative. There's a very large market opportunity. We have robust data, including clinical data and strong IP. And I look forward to meeting with you and discussing Q's platform further. Thanks again for taking the time to listen to Q Biologics oncology focused presentation.